Award for their products from the Standard Organization of Nigeria, SON. In 2016, Dr. Olabode bagged the Master of Science degree in microbiology with specialization in industrial microbiology from the University of Ibadan with a strong academic performance. Dr. Olabode later veered into the vast world of academia to pursue his long-held ambition in the classroom and laboratory. He joined the services of the Federal College of Agriculture, Akure, as a lecturer 3 in 2016. As a result of evidence of his credible contributions to knowledge, dedication to impacting knowledge, mentoring students at both the national and higher national diploma levels, and selfless service to the university community, he rose to the rank of senior lecturer, a position he occupies to date. He obtained a Doctor of Philosophy degree in biology with specialization in storage microbiology from the prestigious Federal University of Technology, Akure, in 2025. Dr. Olabode Efunye, as a seasoned researcher, has to his credit 9 peer-reviewed articles and contributions to global and local academic journals of high repute. He has presented 12 papers and attended several academic conferences and international fora. In addition, he has 2 book chapter contributions to his credit. As a mentor, he has successfully supervised 22 national diploma and 8 higher national diploma student projects in agricultural microbiology and agriculture-related fields. He is a member of the Nigeria Society for Microbiology, NSM, the American Society of Microbiology, ASM, and the Organic Agriculture Professionals in Tertiary Institution in Nigeria, OPTIN. Dr. Olabode has successfully honed his administrative acumen in several college and department positions, which include but not limited to Hall Warden of Deji Mel Student Hostel, Federal College of Agriculture, Akure, Chairman, Research and Development Committee, Department of General Studies, Federal College of Agriculture, Akure, Director, Continuing Education Center, Federal College of Agriculture, Akure. Being a deeply spiritual man, Dr. Olabode Efunye is an ordained pastor in the Redeemed Christian Church of God, where he currently pastors a parish in Obaile, Akure State. He is happily married to the love of his life, Pastor Mrs. Adenike Efunye, and the union is blessed with 2 glorious children from God. Distinguished ladies and gentlemen, I welcome on stage our dear speaker for today's webinar, Dr. Olabode Efunye. Thank you very much.
Thank you very much, Mr. Cletus. I want to really appreciate the African Plant Science Symposium Forum for this honor and the privilege for me to share my thoughts and to interact with you and the vast audience who are here for this webinar this afternoon. It's a privilege I can't take for granted, and I'm very grateful for this. And I hope that at the end of this short interaction, we'll be able to come up with some thought-steering and insightful recommendations, which I know will culminate in your planned symposium for the year. I want to welcome all our audience, and I wish that we will have an interesting, educative, and enlightening time together in this webinar. Thank you once again. I have the duty to interact with you on this important topic, the soil-plant microbe interaction or crop health. And I want to start by stating that at this moment in time, this topic has come at a very, very opportune time, particularly for us in Africa, because it is a matter that has been on the front burner for a long time in Africa with regarding to agricultural productivity and efficiency in the agricultural value chain. I want to say that this topic is very important in the sense that there is a looming threat which all of us have to confront. Now, what is this threat? Presently, United Nations World Population Forum puts human population on Earth at about 8.3 billion people. And with a projection that by year 2050, the world population will have grown to about 9.7 to 10 billion at 0.82% increase per year, increase rate per year. That alone is not a threat anyway. However, it is also discovered that world food production is increasing. But the rate at which world food production is increasing is far, far below and does not commensurate at which the rate of human population is increasing. And this is evident in the challenge of increase in world food prices in the last few years. Now, what has brought about this challenge and what is the threat? The threat is that farming contributes about 85% of human nutrition. And farming is done on land. And that is done to provide nutrition for human population and sustain life generally.
Please, Dr. Alfunwaye.
Yes, sir.
Yeah, your slides are not moving for now.
Sir? No, I haven't moved the slides. I'm just setting a premise for the topic.
Thank you.
Now, the available land for production keeps reducing world over. Why? Because there are many other needs for land, or developmental purpose, and all other things. So for this reason, for us to increase agricultural production and sustain human nutrition, we have to involve ourselves in using a lot of agrochemicals to improve production. And these agrochemicals, because of their synthetic nature, they always leave residue in our environment, particularly in the soil. And many of them over time have become so toxic to this ecosystem. So for this reason, we are having the challenge of inability to meet up with, provide for the nutritional need of growing human population. At the same time, the environment is getting more polluted. So the threat is that in the next few years, we may have the challenge of not being able to feed ourselves, and the land will not be able to sustain it. So this is the reason why we need to confront this with a sustainable means of agricultural productivity. And there is no way we will be able to do this except we understand what goes on in the soil, the interactions between the soil, the plants, and the microbes therein. So that sets the foundation for our discussion this afternoon. And please proceed with me as I go on. When we talk about the soil is the house of plants, and it's already defined as the topmost layer of the earth crust. The soil has by composition minerals which are obtained from weathered rocks, water, air, and organisms, both the ones we can see with our eyes, that's macroorganisms, and the ones we can't see with our naked eyes, microorganisms. The soil is where the roots of plants are anchored. And because the soil is a reservoir of major nutrients needed for plant growth and plant health. The soil is very rich in varieties of organisms, both of which are macro and microorganisms. And these organisms have a lot of roles to play in agriculture. either beneficial or non-beneficial. But for the sake of our discussion this afternoon, we'll be looking at the beneficial roles, beneficial interaction of soil microorganisms with the plants within the ecosystem. The soil is the largest reservoir of microorganisms in the entire universe, and it has the largest microdiversity. So the soil is very important to agricultural practice and productivity. Now, when we talk about the soil microorganisms, like I mentioned earlier on, we'll be looking at the beneficial ones, the ones who interact with the plants, with the soil in a beneficial manner to us. The soil microorganisms are bacteria. You have bacteria in the soil. Bacteria are unicellular microorganisms who may either be gram-positive or gram-negative. We have fungi, some of our which are unicellular, some are multicellular. You have viruses, you have nematodes, which are often macroscopic, and then you have protozoans, who most times are also microscopic. So in other words, most of the microbes that interact with the soil and the plants are the ones we can see with our naked eyes. Well, amongst these groups of microorganisms, the most important and the ones that have been studied extensively in terms of interaction with the soil and the plant are bacteria and fungi. And that is because they play major roles in the biogeochemical cycles. These biogeochemical cycles are the process by which nature recycles nutrients in the environment. So these bacteria and fungi play major roles in this. And some of the beneficial soil bacteria are Azotobacter, Azospirillum, Rhizobium, and fungi, we have Trichoderma, Streptomyces, Fusarium, Pythium. These are some of the ones that play important roles in all these interactions. Now, why are we concerned about plant health? Plants are in ecology, in trophic study in ecology, plants are regarded as producers. Why? Because they are basically the only kind of organisms around us in the environment that have the ability to use nutrients around us in the environment to synthesize, that is to produce food for themselves, and they store some for other forms of life to depend upon. That's why we refer to them as producers. And that underscores the fact that the health of our plants is very, very important to all other forms of life. In agriculture, healthy plants ensure food security and nutrition. In medicine, healthy plants provide basis for most of our drugs, because most of the active agents obtained, that we have in our drugs today, are obtained from plants. In the world of industry, plants provide many raw materials for many of our finished and even semi-finished products that we use. Also in the environment, the plants help us to regulate the climate by absorbing oxygen and by producing oxygen for us to breathe and absorbing CO2, which helps to protect the atmosphere and also prevents us from suffocating. So they help to keep nature balanced. So that underscores the importance of plant health to us. Plant health is important to the health of humans and mostly other organisms. So that is why the interaction of microorganisms with this plant and in the soil in order to promote crop health is a very important topic for us to consider. Now, within the soil environment, there is a narrow plane in which all these interactions occur between the soil microorganisms, and the plants. And that region is what we call the rhizosphere. That is a region in which the plant roots are in contact with the soil. And this region is characterized by the following. One, it has a high concentration of root exudates. What are root exudates? These are byproducts of photosynthesis that plants release through the root cells into the soil environment and usually contain sugars, amino acids, plant hormones, enzymes, and what have you. Now, these root exudates are responsible for attracting the attention of soil microorganisms, because most of the relationship between the plants and the soil microorganisms are symbiotic. That is, I benefit and you benefit. Now, some of these materials that come from the plant root exudates serve as for nutrition for soil microorganisms. And that is why within this region, that is the rhizosphere, is where we have a high microbial population and diversity. This plant rhizosphere, is also the area where minerals and water are uptaken from the soil by the plant. So all our discussion will center around what goes on in the plant rhizosphere. The characteristics of a plant rhizosphere is determined by the nature of the root exudates that it releases. And so the microbiome, that is the microorganism found within a soil environment, is determined by the kind of exudates that the plants release into such an environment. And like I mentioned at the beginning, we will be looking at the beneficial roles of these soil microbes. I mentioned that there are bacteria, fungi, that is mycorrhiza, that is fungi that are found in the soil that interact with plant roots, fungi, viruses, protozoans, and some algae in aquatic environment. What are the major activities they carry out? They carry out decomposition of organic matter, complex organic matters, into simple inorganic matters that the plants can easily take up. They also carry out the activity of fixation of some of the nutrients for plants to grow and for crop health. They help to solubilize some nutrients, and then they also help the plant to defend itself against stress condition. Please permit me to just go through some of the benefits that plants derive from activities of these microorganisms. They make nutrients available. They enhance the uptake of nutrients from the soil and water by the plants. They regulate the biogeochemical cycles. They degrade macromolecules that I mentioned earlier on. They also help plants to resist some biotic and abiotic stress in the soil environment. And how do they go about this? Let's quickly take a few. Now, some of these activities are direct. They relate directly to plant health and plant productivity. The first one we'll be looking at is mineralization. That is the breakdown of organic matter into simple inorganic minerals, which are utilizable by plants. Now, these organisms help to break down large organic materials, dead animals, plant debris, and other organic wastes in our environment. They help to decompose them and break them into simple inorganic matter that can be assimilated in solutions in the soil. So they make these nutrients available to the plant roots right there in the rhizosphere. Now, examples of such microorganisms are fungi. They are the major decomposers of macroorganic molecules into small. And I have quite a number of them: trichoderma, rhizopus, aspergillus, and what have you. You also have some bacteria that carry out these functions too. They help to break down allocotinous and autocotinous materials within soil environments. And then some protozoans play this role as well. Then another direct interaction between soil microbes and plants is in helping to fix nitrogen. The importance of nitrogen to plant growth and health is very immense. Now, nitrogen, the soil is not very rich in nitrogen, but the atmosphere is very rich in nitrogen. In fact, by composition, air is about 73% nitrogen. So there are some soil microorganisms that have the ability to convert this nitrogen, atmospheric nitrogen, into simple forms like ammonia. They reduce, they have to oxidize the atmospheric nitrogen into ammonia, which the plant cell can easily absorb from the soil. Some go further to oxidize ammonia into other soluble forms like nitrites, nitrates, which dissolve in soil water, and this plants will be able to absorb them through the root hairs and use them for all the nitrogen is used for in plant growth and plant health. For example, it's very important that, nitrogen is very important to chlorophyll formation in plants. And we know that chlorophyll drives photosynthesis. So these organisms play a vital role in making nitrogen available in natural form to plants in the soil, which now reduces our dependence on chemical fertilizers, that is our common nitrogen fertilizers. So the more of these organisms and their activities and interaction with the plants in the cell enriches the soil with nitrogen, which reduces our dependence on chemical and help to protect the soil, even as it improves soil crop health. Nutrient solubilization is another function, direct function of these organisms in the soil. They help to release minerals such as potassium, phosphorus, zinc, which most times occur in the soil in a form that are not accessible to plants. Some of these compounds, they are found in the soil in complex forms, which these plants cannot assimilate. So these organisms help to solubilize and bring out these nutrients from these complex forms and break them into simple units. So thereby making these important nutrients available to plant soil. Examples of such organisms, we have them there. Pseudomonas is very active in that. Fibobacterium is very active in that. As well as Azotobacter, Azospirillum, they play very important roles in that. And they do this through many means. Some of them have the ability to synthesize, some of these organisms have the ability to synthesize organic acids. Organic acids help to reduce the pH of the soil, which helps to solubilize these nutrients out of the complex forms, which they are, into simple inorganic forms that the plants can use. Nutrient mobilization is another direct function of this. In interacting with the soil, they help the plant to uptake some of these nutrients far quicker and more efficiently because of their interaction. For example, most times the bacteria will solubilize the nutrients, while the mycorrhiza, that is the fungi that are in association, symbiotic association with the plant roots, either inside the plant root or outside, ecto or endo-arbuscular fungi. They help to bring these solubilized minerals into the plant roots here and for easy assimilation and translocation to other parts of the plants. Another direct role that these microbes, by their interaction with the plant, is to help in producing plant growth promoting substances like plant hormones like auxin, gibberellin, ethylene. They help to promote growth in plants. The presence of these microorganisms most times helps to regulate the production of these plant hormones and other substances like indo-3-acetic acid, salicylic acid, and cytokinin, which plays vital roles in the productivity of crops. Some of these plant growth promoting bacteria in the soil are Pseudomonas putida, which have been used extensively in many studies. Azospirillum, Lycophorum, Bacillus licheniforms, these have been used in studies on many crops, maize, beans, mung beans, and all that, and have shown the ability to help to promote plant growth. when compared to plants that were not inoculated or that do not have these organisms within their soil environment. So microorganisms, soil microorganisms help to regulate the synthesis of these plant growth promoting substances in plants. Another direct benefit of soil microbes in their interaction with the plant is indirect now. Now, indirect mechanism of promoting plant growth and health by this microorganism is by acting as biocontrol agents. When acting as biocontrol agents, that is, agents that have the ability to inhibit or control the growth of plant pathogens. When we talk about plant pathogens, we're talking about plants that have the potential to cause diseases. Organisms that have the potential to cause diseases in plants. And we have them in the soil. So the presence of some of these beneficial microbes helps to control the growth of some of these, either by production of antimicrobials like phytoalexin, camalexin, and even itoin A, which is produced by Bacillus subtilis in controlling some soil pathogens like Fusarium, Pythium, and water beetle. They also do this by parasitic actions, that is, eating up other microorganisms. Amoeba Proteus does that a lot, feeding on pathogenic bacteria in the soil, thereby reducing their population and helping the plant to be healthy. Some of these microorganisms also help the plants to boost their immune system in very many ways, which helps them to withstand the presence of biotic pressures like these pathogens. Another indirect way is the ability of these soil microorganisms by their interaction with the roots of plants. They enable the plants to tolerate some abiotic stress, environmental stress, like drought, high salinity in the soil, and at times exposure to heavy metals in the soils as a way of our human activities. Some of our industrial and even domestic waste, we just dump them indiscriminately, and then they find their way into the soil, and they contain heavy metals which are assimilated in the soil, absorbed in the soil like that. So the presence of some of these soil microorganisms help the plants to tolerate and to withstand these environmental stresses. For example, when a plant is subjected to drought stress, the plants produce a compound known as 1-aminocyclopropane 1-carboxylic acid. Now, the accumulation of this substance in a stressed plant would trigger the production of stress ethylene. Ethylene is a plant hormone. This plant hormone, ethylene, accelerates the rate of aging in plants. So when this stress ethylene is produced in plants as a result of drought stress, it causes death of shoots and roots of plants. However, the presence of some beneficial microorganisms like Pseudomonas putida or fluorescens or Variola paraxylus have shown their ability to help plants to tolerate this condition. And how do they do this? They produce an enzyme, ACC deaminase, which helps to break down the accumulated ACC, that is 1-aminocyclopropane 1-carboxylic acid. They break it down into simple forms that the plants can now use as nutrients. So this helps the plant to withstand drought. Similar way is done in high salinity soil environment. Some bacteria have the ability, and even some fungi have conferred the ability to break down this accumulated stress compound into simple compounds like alpha ketobutyrate, even ammonia in some cases, which will help, which are which serve as nutrients to plants, even in such stress conditions. Some other plant growth promoting bacteria will also produce siderophores, which help to liberate metals, because in high salinity environments, the metal nutrients are locked in the soil. So the plants do not have the ability to access them. So the presence of these microorganisms production of siderophores will help to liberate the locked in metal nutrients like potassium, phosphorus, and bring them into form of accessibility to root plants, which will now enable the plants to uptake them and use them for growth maintenance. Another way by which these plants growth promoting organisms, helps plants to tolerate high salinity conditions, is by increasing the osmotic potential inside the cells of the plant themselves to withstand water loss by osmosis to the environment, thereby producing amino acids like a proline and soluble sugars, which the plant root hairs assimilate into their cells, thereby increasing the osmotic potential in the cells and withstand dehydration that comes around by high salinity. So these are some of the mechanisms by which these organisms help plants to tolerate abiotic stress, promote health and productivity in health in challenged environmental conditions like this. Now, having seen both the direct and indirect activities of soil microorganisms in interaction with the plants in the root cell, a lot of study have led to the development of bioinoculants. Bioinoculants are microorganisms that are specially developed for application on seeds, plants, and even for mixing into the soil, which confers some benefits on the plants and the soil. When you introduce these organisms, they help the plant to grow better when compared to when they are not introduced into the soil or introduced into the plants. You may inoculate them into the seeds or seedlings directly. For example, in 2021, Canning and his co-workers, they were able to improve the growth performance in maize plants. That was in Malaysia, by inoculating a consortium of bacteria and fungi into the substrates upon which the plants were grown in potted plants. And they discovered that the potted-based plants had better root development, shoot development, and higher number of leaves than plants that were grown in soil that were not inoculated with this bio. So that tells you the importance of bioinoculants. They are now available in commercial forms that farmers can easily access and use. The effectiveness of these bioinoculants now depends on the consortium of microorganisms and the carrier materials in which they are immobilized, because the organisms are the natural carriers include carbohydrates like cellulose, starch, paginates, which are very good substrates to fix these microorganisms in. They help to protect the microorganism. They help to provide an environment suitable for viability of these organisms until you inoculate them into the soil or into seedlings. However, there are some other synthetic carriers that are used in producing these bioinoculants like polyacrylamide and polyurethane. But the choice of natural carriers is much more preferred, in the sense that these natural carriers most times are biodegradable, and then they help to sustain the microorganisms far better than synthetic carriers. So bioinoculants, they play a very important role in the present day agriculture in order to increase soil fertility, improve crop health, and productivity. Now, the discovery and the development of bioinoculants has also generated new innovations. We now have bioinoculants enhanced, which we refer to as biostimulants. Biostimulants contain these plant growth promoting microorganisms with plant hormones, plant enzymes, trace elements, and seaweed extracts, which enhances and improves the activities of these microorganisms in these preparations. And I must also mention that the use of bioinoculants is very easy for farmers. You have them in liquid form. You also have them in solid forms. So they are made easy for farmers to apply. Another innovation that has come on board, which is being studied extensively now, is bio-nanotechnologies, where we inculcate nanoparticles into plant growth promoting microorganisms to enhance their desirable activities when introduced into plant soil environment. So I mentioned earlier on, all these new innovations are eco-friendly, they are effective, and they are also user-friendly. When we talk about soil micro-plant interactions, how do we optimize our knowledge of this? The development of bioinoculants leading to nanotechnology products, biostimulants, came as a result of the introduction of new scientific techniques in studying the interaction between soil microorganisms, and the plants within the rhizosphere. And these new techniques has really enriched our understanding of this complex interaction between these three components within the environment. And these techniques include metagenomics studies. Now, in metagenomic studies, we are able to extract all the genomes of all the microbial community within the soil environment and profile them. Only about 30% of microorganisms in the soil can be cultured outside of the soil environment. That is, can be cultured in the laboratory. More than about 70% are still not unculturable. So studying them in the laboratory has been a challenge. But by introduction of metagenomic techniques, we can determine the entire genome of the microbial community there, and then understand the complex interaction between the soil, the microorganisms, and the plants. So this has really helped. However, it has its own limitations, and that is why another technique developed, which is metatranscriptomics, which is the use of transcribed gene sequences to understand the complex interaction going on in the soil environment. This technique do not profile DNA segments or fragments, but it profiles the messenger RNA that were produced from transcription of the active DNA segments. This gives a better understanding of which of the genes from either the plants or the microorganisms in interaction was actually regulated to transcribe into messenger RNA. So profiling the mRNAs give us a clearer understanding of this complex interaction. However, it also has its own limitations, thereby giving room for the development of metaproteomics technique, which studies the proteins that were translated from these messenger RNAs. What are the proteins that were produced within the microbial community? We're able to profile them using a lot of genetic tools. And then the fourth technique is metabolomics, where we study the metabolites produced by all these components in the interaction. That is what are the what are the plant metabolites produced in interaction with the soil microorganisms? What are the metabolites produced by the soil microorganisms in interaction with the plants? So having an understanding of the metabolites gives us an indication of the kind of interaction that is occurring amongst soil microorganisms and between them and the plant roots. So the combination of all these genetic techniques has come under the broad view of multi-omics approach to studying the complex relationship or interaction between the soil, plants, and the soil microorganisms. So it is the aggregation of all this that led us to the development of a tool now being used to develop inoculants. And that tool is called synthetic biology. Synthetic biology is simply a procedure that was developed to produce the desired influence on the microbiome genome using gene editing tools. Having understood the genes that influence the interaction between soil microorganisms and plants, in plants and in microorganisms, we're able to now use genetic tools to edit these genes in order to improve or enhance certain desirable characteristics in either the plants or the soil microorganisms or even the both together. So that approach is what is being studied extensively in research into soil, microbe, and plant interaction. And we are able to optimize that, what is an ongoing work. So having done all this so far and the progress that have been made and the successes that have been recorded, there's still a lot of work to do. So what are the future directions in studying and understanding the soil, plant, and microbial complex interaction? We need to have a deeper understanding of the non-culturable soil microbiomes, which I mentioned earlier on. More than about 70% of the microorganisms in the soil are still not yet properly understood. So there's still a lot of work that's still being done in this area. Then the interaction between the indigenous soil microbiome and the bioinoculants being introduced is also an area that is still being studied, because each soil environment has its own microbiome, that is the native, if I permitted to use that word, the indigenous microorganisms within that soil environment. When you now bring your bioinoculants into that environment, how do they interact with this indigenous microbial community? How do they interact with the cell? Are we with the plant? What kind of interaction? Is it synergistic? Are there antagonistic interactions? So these are still areas that are still being studied. So we also need to study further the effects of our common agricultural practices on our commercially viable bioinoculants, and also the effect of global climatic change on the commercially viable bioinoculant, because climatic change is a big factor in agricultural productivity today. How does this affect the development of bioinoculants? So these are areas that we are still looking at, and a lot of work are still going on in these areas. So I would like to conclude that soil, microbiology, and plant interactions are complex and may either be beneficial or adverse. The beneficial soil microbes play their major roles in the fixation, solubilization, and mobilization of nutrients in the soil in order to aid nutrient efficiency and improve crop productivity by enhancing crop and soil health. This Soil microorganisms that are beneficial also enhance the resistance of plants to both biotic and abiotic stresses in the environment using various pathways and mechanisms. So our understanding of the soil microbiology and microbe plant interactions has improved tremendously over time. And this has culminated in exciting technologies, which I've mentioned earlier on, that have helped us to develop effective biofertilizers, which has shown varying degrees of successes in the laboratory, in the field, and large-scale applications. So this enables us to will enable us to practice responsible and sustainable improvement in food production to attain food security even as global population is increasing. So, a way of recommendation, more advances needed into microbial ecology technology, and that must be encouraged across research institutes and universities. And when I talk about my country, this area needs a lot of funding in Nigeria because in Africa, we are agrarian community. So this requires a lot of fund support for research and advance into this area. Also, fund investment is also needed in the production of biofertilizers by all levels of government and private organizations to make them much more available, accessible, and economically viable for farmers. Government policies also needs to promote sustainable agriculture in order to reduce our reliance on synthetic fertilizers, in order to protect our environment from pollution. Finally, advocacy among farmers must be embraced. To embrace the use of biofertilizer is very important because generally studies have shown that farmers are not always open to introduction of new techniques or new technologies. There are always cultural bias against this. So a lot of advocacy work will also need to go into that in order to help us drive agricultural productivity, sustainable agricultural productivity, improving interaction between the soil microbes, the plants, and the soil in Africa. Thank you for this opportunity. Thank you. I appreciate you. Thank you.
Thank you, Dr. Olabode Ifumoye, for your nice presentation. I'm Abdul Muiz Salau, and I will be the one taking us through the Q&A session. So far, we have had 2 questions in the chat box. So
Okay.
the first one is from Naman Arodi. He or she is asking, how available are these biofertilizers to small-scale farmers, and are they affordable?
Thank you very much. They are available. However, just like I mentioned earlier, a lot still needs to be done to downscale. Yes. In the sense that they are available, but they come at a reasonably high cost because production level is low and the demand is increasing because of the increased awareness in organic agriculture. So it's a big challenge, and that is why I advocated and recommended